So today, in my talk, I'm not mentioning fibrin, I'm not mentioning TPA. I'm going to be focusing on urokinase and its role in pancreatic cancer specifically, growth and metastasis, and some of the mechanistic things we're getting to understand what's going on. This crowd, just to refocus you away from TPA and fibrin, in cancer, UPA and its receptor UPAR is upregulated extensively. It's a real thing here, and it's important in activating co-localized plasminogen, and we all heard about the various plasminogen receptors that can be available in various cell types. And this co-localized plasminogen is efficiently activated to plasmin. And plasmin can either directly cause tissue barrier breakdown or ECM breakdown to lead to invasion and metastasis, or it can indirectly do so by activating pro-MMPs or activating latent growth factors in the ECM. We recently did a review of all the literature, of all the stuff involved, all the UPA papers involved in pancreatic cancer specifically, both experimental work and clinical work that shows that the UPA system is upregulated. It does affect pancreatic cancer pathogenesis, and it's a prospective diagnostic and therapeutic target. Some earlier work that we've done using this International Cancer Gene Consortium for the PACA, the Pancreatic Cancer Group in Australia, we show that high PLAU or UPA mRNA levels, the higher you have this, the worse your prognosis. And this is in patients who had resectable pancreatic cancer. So there's 20% of patients who present who can still have their tumours removed because there's no evidence of locally advanced disease, still have poor prognosis, and the higher the UPA, the worse their prognosis. And this is using the Clinical Proteomic Tumour Analysis Consortium and looking at protein levels, and we can see also that high UPA gives a worse overall survival outcome for these patients. So it correlates with worse clinical pathological features and poor prognosis in PDAC is pancreatic ductal adenocarcinoma, which is 90% of all pancreatic cancers. So suggesting that targeting UPA would be really a good measure in pancreatic cancer, because there really aren't any good treatments for advanced disease. And pancreatic cancer is one of the worst prognosis of all cancers. So you'd think that, well, there have been a lot of people developing UPA-targeted drugs over the years, and only one has gotten to a clinical trial, and this is this upamostat, now owned by Redhill. This is the structure of it, small molecule inhibitor of UPA. And they did show in a phase 2 trial on locally advanced non-resectable pancreatic cancers that there was a 17% increase in 1-year survival in combination with gemcitabine, which is a commonly used chemotherapeutic for pancreatic cancer. It was an improvement over gemcitabine alone. But they have since stopped doing any cancer-related trials because really this inhibitor is actually a broad-spectrum serine protease inhibitor. It's most active against trypsin isoenzymes and has multiple. They now are marketing it as having multiple indications, and one of them is for use in COVID. So there's no more trials in cancer because it's not specific enough, I think, for cancer. So we believe we need a more selective UPA inhibitor for cancer for this purpose. So our approach derived from a medicinal chemist who approached me, who was working on various small molecules, including amiloride for other things, as a potential lead UPA inhibitor. Amiloride has been around for a long time. It's a diuretic anticholinergic. It works via epithelial sodium channels. That's its main effect, but it has got this side activity of inhibiting UPA with moderate effectiveness. It's got moderate UPA selectivity. Will target other trypsin-like serine proteases. But the problem with using amiloride as an anti-cancer drug, and people have done that in animal models, is that the maximum tolerated dose for a human is 20 milligrams per day, and that is not high enough dose to be used as an anti-tumour agent. Any higher than you have hypertension problems, cardiac arrhythmias, and high blood pressure issues and kidney damage. So it can't be used in this way. So he said, can we use this as a scaffold to improve the selectivity for UPA, remove the ENaC activity, so we can give higher doses and also make it more selective. So with a couple of PhD students, and Ben Buckley was one of the key ones, multiple iterations, structure-activity relationship studies, we came up with maybe 50-odd analogues of amiloride, and the 6-hexamethylene amilorides had inhibitory constants in the 50 to 500 nanomolar range, which is pretty potent. Out of those, we settled on this particular compound, a paramethoxymethyl group, BB230F, that's Ben Buckley too, one of his 30th one. And we followed this up and studied that in detail. It is highly selective, so it's got no activity against other trypsin-like serine proteases, and we did a much bigger panel than this. So that was good. It lacks the human epithelial sodium channel activity and other ion channel activities, because amiloride does inhibit other ion channels, including A6 and NHE1. So it doesn't do that, so we're really looking for something on target, no off-target effects. In work done by a current PhD student, Ashna, we've shown using cell lines that we're using in our models that it's not cytotoxic, only when you get to really, really large concentrations, you see some signs, and it's well tolerated in vivo up to 10 milligrams per kilogram in rodents, and it efficiently inhibits cell surface UPA activity and subsequent plasmid activity, and that's really important that you're inhibiting the target on the cell surface. So, okay, we did a whole bunch of biochemical stuff to show the and PK analyses to show that it's got reasonable drug-like properties, not the best, but it's okay, and that it really is selective. So now, to my mind, we needed a really, really good in vivo model to test the hypothesis that inhibiting UPA inhibits really metastasis is what I was looking for, but of course, if it has effects on the tumour growth, invasion and metastasis, that's great. So then I turned to a colleague, Professor Minotti Apti at the Ingham Institute and UNSW, who had developed this very nice orthotopic model, which means placing the tumour cells directly into the pancreas, using co-implanting them with the supporting cells that are found in the pancreas, the pancreatic stellate cells, and they're responsible for making the collagenous stroma that is present, it's plastic stroma that's present in pancreatic cancer. So we have a xenograft model, so we use human cancer cells or human-associated human pancreatic stellate cells, or a syngeneic model, we have KPC-derived tumour cells, the KPC mice, or murine stellate, pancreatic stellate cells. And they're implanted orthotopically in a 1-to-1 ratio. And what we find is that these models both give robust primary tumour growth by 7 days post-implantation. They resemble human PDAC, pancreatic ductal adenocarcinoma, by having this characteristic desmoplastic stroma, it's very collagenous. And these mean that they're clinically relevant. And they also have high UPA and plasmid activity. So this is UPA activity in the tumour tissue versus normal pancreas and plasmid activity, which is what we see in locally advanced and metastatic human disease. And these models give 100% spontaneous metastasis. That's really important. We really wanted to have models that we know for sure these mice are going to get metastasis. So then one of her PhD students working with us did these treatments on these models. He did what we call an early intervention, which is treatment starts 1 week post-implantation, so it's sort of equivalent to a resectable model, or advanced intervention, where the tumours were allowed to grow for 2 weeks for the syngeneic, because they go really fast, or 4 weeks for the xenograft model. And then the tumour size is confirmed by ultrasound to make sure that they're there, and then the mice are randomised. And then and that's equivalent to an advanced metastatic model, this advanced intervention model. And then the treatment starts for 2 or 4 weeks, depending on the model, with control or gemcitabine at this dose, which is equivalent to human doses biweekly, or BB230F, just shown here as UPA inhibitor U, at 10 milligrams per kilogram per day, or the combination. And then treatment starts at 2 to 4 weeks, treatment goes for 5 to 6 weeks, and then euthanasia and collection of tissue, and assessment of tumour size, metastasis, and histological analysis. And we looked at these parameters in the primary tumour. This time is the tumour immune microenvironment. So we looked at all of these, and this is just showing some mets. So to summarise a lot of data, so what we see here in the xenografted or the syngeneic, which is an immunocompetent model, is that you have robust primary tumour growth, and then gemcitabine has some reasonable significant effect on inhibiting the primary tumour, as does the inhibitor, the UPA inhibitor, 30F alone, and the combination is good and better than gemcitabine alone. And we see similar effect in the syngeneic model. So that's on primary tumour growth, which is great, but what happens in metastasis, against metastasis? And what we see, at least by what we can see with visible metastases and by doing cross-sections and histological staining, is that, 9 out of 9 mice see with at least 1 liver metastasis. The gemcitabine is hopeless in inhibiting metastasis, and that's known. It's not unknown. So 30F alone is pretty good at eliminating metastasis, and the combination wipes them out. We didn't see any. And that happens in both the xenografted and the immunocompetent model. And we did look across a whole bunch of other tissues to look at metastasis, and just highlighting that in the 30F group and the 30F plus gem, you basically see a lot of zeros, meaning we really couldn't see many or any metastases anywhere compared to controls and the gem group. So I won't show you all of this data. You're going to take my word for it because there's a lot there. We looked at all these parameters in the primary tumours, and we found that in the primary tumours, proliferation, ECM, markers of ECM degradation, markers of stemness, and markers of epithelial mesenchymal transition were all decreased in the primary tumours that were treated with 30F plus or minus gem. Sometimes 30F did all the heavy lifting, sometimes we needed the combination. But in the syngeneic models, we also found that the 30F in particular promotes an anti-tumour immune response. And just to show you some of that data, so we saw that there was a trend towards an increase in total T cell infiltration, but a significant decrease in the CD4 positive cells, mostly due to the 30F, and a significant increase in the cytotoxic or CD8 positive cells. The combination here was better than the UPA inhibitor alone. And a significant decrease in M2 macrophages. These are a decrease in the pro-tumour type macrophages, and a decrease in TGF-beta, very significant, which also is responsible for immunosuppressive environment. And remember, maybe I did not mention that PDAC is unresponsive to immunotherapy. They're considered immunologically cold tumours. So potentially this kind of treatment may actually make them responsive to immunotherapy as well. Ashna has also been using this tissue localization technique using matrix-assisted laser desorption ionization mass spectrometry imaging. It's a label-free analysis of and localization of endogenous metabolites and any small molecules you might be adding to the system. We can see in this mass to charge range, that we can find our BB230F. This is molecular mass. And it's absent in untreated tissues. We can also look at a whole bunch of other metabolites, like lipids, as you'll see. She also used MSMS as a secondary validation to see that we are indeed picking up the 30F. This is a drug standard, and these peaks arise after the laser treatment of the drug. It breaks it down a little bit. And what we see here, we can absolutely see in the treated tissues, just one representative example, 30F, which is yellow, in the periphery of the tumours. And there is, in this example, some necrosis, which when you overlay with the H&E, which is done after the mouldy MSI, we can see that the distinct co-localisation of 32F in the tumour-rich areas and near the tumour periphery. These are different types of lipids, and she's looking into what the lipid profile looks like also in response to 30F. So what are the underlying mechanisms? We know that there's a sort of crosstalk between the stellate cells and tumour cells. They both kind of co-activate each other. And a result of the effect of stellate cells on pancreatic cancer cells is to affect a whole bunch of physiological processes that result in increased cell survival by increasing proliferation, decreasing apoptosis, affecting EMT, so increasing invasion and migration, and increasing stemness. And that 30F somehow is interrupting this process. So we did a bunch of co-culture studies using this transwell or Boyden chamber type arrangement with the stellate cells on top and the tumour cells underneath. This is the human system, so they can crosstalk by with soluble mediators, and then look at various parameters. And in this case, we're looking at proliferation. So these are the cells, the tumour cells, in the presence of the stellate cells that enhances their proliferation as expected. And then gemcitabine alone decreases that proliferation, as does the 30F alone and the combination. Okay, that's good. So we know we get an effect of proliferation, but it's probably gem doing the heavy lifting here. With apoptosis, what we see is in this assay, the tumour cells are serum starved to enhance apoptosis. So most of the cells here are apoptotic in the tumour cells. And when we co-culture with the stellate cells, that decreases the number of apoptotic cells, so it improves their survival. Gem that has no effect on this. And these, I should say, these are doses used that are not subcytotoxic, by the way. 30F alone really reverses that apoptotic effect. So it normalises the situation so that cells now undergo apoptosis. And in the presence of gem, the effect is not different to just UPA alone, a UPA inhibitor alone. And we looked at cell migration, basically similar setup. The stellate cells enhance migration, and gem inhibits that, as does 30F, but the combination here works much better. And lastly, we looked at some cell signalling pathways, and gem itself doesn't do anything, but the inhibitor, the 30F inhibitor, does decrease signalling via MAPK and also by PI3K, which is not shown here, pathway. So in conclusion, I think I've shown you that, hopefully I've shown that selective UPA inhibitor 30F is anti-metastatic and reverses the immunosuppressive environment. And it could do this by just directly inhibiting the formation of plasmin, which then will inhibit these downstream processes. But remember, plasmin can also activate latent growth factors, so maybe by inhibiting this process, growth factors are not released and activating the system. But also, you may or may not recall that there is a body of literature that says that UPAR can signal indirectly via interactions with integrins. So there could be some signalling things going on down here that interferes with. the complementary in vitro co-culture study suggests that BB230F disrupts tumour cell, stellate cell crosstalk. And a hypothesis we have, because my colleague Minotti has shown that if she uses a similar in vivo system and puts in anti-HGF antibodies or anti-c-Met, which is c-Met is present on the surface of cancer cells, HGF is secreted by the stellate cells. If she puts in anti-HGF or anti-c-Met, she sees a similar effect that we see on all those parameters by inhibiting UPA. It's known that UPA, at least in vitro, can activate HGF precursor to active HGF. So it's a hypothesis we're chasing that this 30F could be interfering with this conversion of inactive growth factors to active growth factors, and that has downstream effects on all these cellular behaviours. But in the end, I think this 30F experiments, I think, confirms a causative role for UPA-mediated mechanisms in tumour invasion and metastasis, and these are, at least in part, go through tumour stromal cell interactions, and it validates UPA as a target for development of drug candidates for clinical use. And with that, I'd like to put up the most important slide, as people have been saying today, and acknowledge all the people involved in this work. And thank you.